So we're gonna begin with this presentation session right now. The first thing we're gonna watch is iGEM Glasgow. So you already know, go for it.
Hi, we're Glasgow, this year's iGEM team from Glasgow. Our team is made up of 7 molecular biologists, 3 engineers, and 1 computer scientist. We have 2 supervisors and 2 advisors, and this is Vita, our mascot. He was part of our outreach. I'll first give you some background information about our project. Nutrition is an important part of health and development, and generally, better nutrition equates to a better life. Sadly, adequate nutrition is a problem around the world, especially in low to middle income countries. Malnutrition contributes to almost half of all child deaths under the age of 5, and vitamin deficiencies can have detrimental effects. For example, blindness is caused by deficiency in vitamin A, which is what we focused our project on. Vitamin A is important for the vision and the immune system, keeps the skin healthy, and is required to maintain specialized epithelia. Around 1/3 of the world's preschool age population is estimated to be deficient in vitamin A. This can cause a number of problems in the eyes, which can ultimately lead to blindness. Although this deficiency has been recognized as a problem, the current solutions are quite limited. For example, massive dose supplementation is an injection of a high dose of vitamin A that is administered to children every 6 months. Not only does this provide discomfort, but the effects wear off already after 2 months. Golden rice is an example of food used for supplementation, but its development has been quite difficult. So we've come up with our own solution, and that is beta-carotene enriched yogurt. Yogurt is a staple food around the world, including areas where vitamin A deficiency is prevalent. We contacted an expert in South India, and he informed us that a large number of households in India make their own yogurt every day. So we thought we'd take advantage of this. So the aims of our project were to engineer yogurt bacteria to produce beta-carotene. We also wanted to characterize these yogurt bacteria to improve the ease of use in biotechnology. We wanted to design a system to prevent the growth of the modified bacteria outside of the yogurt environment, and we wanted to create a piece of accessible hardware to enhance yogurt production in low-resource settings. So Streptococcus thermophilus is one of the main bacteria used to make yogurt. We made this our main target organism, but we initially worked with E. coli, as it's well characterized and easy to work with. We designed a shuttle vector to transfer our constructs between these 2 bacteria, which Matt will talk more about later on. We made a 4-gene biobrick construct that allowed E. coli to produce beta-carotene, the precursor of vitamin A. We chose to synthesize beta-carotene rather than vitamin A, as excess vitamin A is toxic to humans, and the body can just convert beta-carotene into vitamin A as needed. This is part of the beta-carotene biosynthesis pathway. Streptococcus thermophilus already has the precursor GGPP, so we needed to include CRTB, I, and Y to complete the pathway. For E. coli, we needed to include CRTE to allow it to synthesize beta-carotene. We thought this would be a good idea to include in Streptococcus thermophilus as well, as it would increase the concentration of precursor molecules made, and therefore increase the yield of beta-carotene. So that here is our biobrick construct with the beta-carotene genes, and as beta-carotene is orange, the orange pellets here show that we've qualitatively made beta-carotene in E. coli when compared to the white control pellet of E. coli. We then quantified the beta-carotene in our construct. We did this by acetone extraction. So we measured the absorbance spectrum of the acetone fraction, and as you can see, it's quite similar to the literature spectrum, so this indicated the presence of beta-carotene. We then measured the absorbance at 454 nanometers, which is the peak of the absorbance spectrum of beta-carotene. The top 10 E. coli cells were used as a control to show the background absorbance, and here you can see that we have successfully made beta-carotene even without a promoter. And this is probably due to read-through transcription of the vector. So for further work, we want to investigate other genes in the pathway and see how that affects beta-carotene synthesis. We'd also want to use a known mass of beta-carotene to create a calibration curve so we know exactly how much our sample has. We'd also want to estimate the beta-carotene levels in the standard yogurt portion, as well as golden rice, and see how that compares to our product. We'd also want to compare beta-carotene levels in our yogurt to the daily requirement values. I'll now pass you on to Matt, who'll talk about Streptococcus thermophilus as a chassis organism.
So, as Kenny already mentioned, our target organism for this project was Streptococcus thermophilus and not E. coli. Now, the reason for this is that while E. coli is a very useful model organism, and has been for several decades, and I'm sure most of you have actually worked with it cannot produce yogurt. It just lacks the pathway needed to ferment lactose into lactic acid. However, S. thermophilus has sort of the reverse problem, where it's very good at producing yogurt. In fact, it's necessary, but it's not all that well characterized. There are only maybe a few labs in the world that work with it and actually know how to manipulate it correctly. And what this means, especially in the context of iGEM, is that we lack the very kind of standardized parts of biobricks with known outputs and behaviors that can be used in Streptococcus thermophilus, which lacks these kind of parts. So from the very beginning of the project, we set out to have a sub-project focused on turning Streptococcus thermophilus into a chassis. That is, laying the groundwork so that not only could we work in it more easily, but also so that any future teams which want to work with either yogurt or cheese production could actually also use what we worked on and further it in the context of biotechnology. One very interesting thing about Streptococcus thermophilus is that it is naturally competent. So as you know, E. coli, for example, requires either calcium chloride or electroporation to become competent and thereby take up DNA. However, Thermophilus has an inbuilt pathway which allows it to take up DNA under certain circumstances. So this is here, I'll briefly take you through it. So top right you have COMS, which is a small peptide that the bacterium synthesizes. This is then recognized by a receptor in the cell called COMR, which binds to COMX, promoter, which then goes on to transcribe genes that actually induce proteins. So we thought of this as a very nice alternative to the current process of making bacteria competent, and we want to further this and see if we can actually make it work in the context of iGEM. So provide very standardized protocols for future teams to work on. So there were basically 3 steps in our process of turning S. thermophilus into a chassis. The first one was identifying and standardizing a shuttle vector. Shuttle vector basically means that it's a plasmid that can work in both E. coli and S. thermophilus. And so this allows us to perform gene assembly in E. coli, which as we all know is easy to work with, but then transfer our final product into S. thermophilus for actual expression and implementation. So to this end, we acquired a plasmid called PMG36CT, which has a broad host origin replication. And we basically turned it into a biobrick compatible plasmid backbone by taking advantage of a few inbuilt restriction sites to insert the biobrick briefing suffix. So this has actually been submitted as a part, and any future teams can basically use this to just put in their own biobrick parts into it and transform either E. coli or S. thermophilus, as we already did. The second part of the chassis was actually identifying promoters that can actually work in S. thermophilus. And the reason for this is that S. thermophilus is a gram-positive organism. So what this means is that there's no guarantee that very well characterized promoters, such as the Anderson promoters, will work within it, even if they work within E. coli. So to basically identify a few promoters that will work in S. thermophilus, we did a brief literature search to identify a few of them, and we picked 3: P25, P32, and PHLDA. Now, we knew that all of these were constitutive promoters, but we didn't actually know just how strong they are. So as a preliminary test, we fused them to GFP as a reporter, and we did a kind of qualitative analysis of just how strong they are. So you see here at the top right, we have the order. PHLDA turned out to be pretty strong. It has a very kind of intense yellow color, whereas P25 is slightly more faint. Where this is really just qualitative analysis. And what we really want to do was find out just how strong they are and how they compare to each other in relative terms and percentages. So to this end, we turn to the well-characterized Anderson promoters, which are just a family of constitutive promoters used in synthetic biology. We fused all the promoters to GFP as a reporter, and we grew them up in E. coli cells and used a plate reader to quantify fluorescence. So this yielded a pretty nice graph. So as you can see here, the Anderson promoter bars are the blue bars, whereas our S. thermophilus promoters are the yellow ones. So all the way on the left, you see J23100, the strongest Anderson promoter, which is basically the benchmark for strength. And you can see that 2 of our promoters are actually relatively strong. They're about 80% to 90% as strong as J23100. However, we also have P25, which is slightly more moderate, coming in around 40% strength. So what this gives us is a nice functional range of promoter strength. So we have, if expressing a protein too highly kills the cell, we can turn to P25 instead. The next step, and the final step, because so far all the work had actually been done in E. coli, was physically transforming Astromophilus. And to do this, we used the COMET system I mentioned previously, and we decided to use AMLCP, a blue chrome protein, as a visual reporter. And the reason for this is that Astromophilus grows sort of slowly. So having a very kind of quick assay to see whether it's been transformed or not is very helpful. So we fused our promoters to AMLCP and tried to make this work. However, this turned out to be a bit more difficult than we thought. It was far from straightforward, and in fact, the bulk of our project was fiddling with values to try to make the transformation work. And in the end, however, this did pay off, because we did manage to transform both E. coli and Astromophilus with this plasmid. We didn't only get Astromophilus colonies, but we also got, we also mini-prepped these colonies, and then retransformed E. coli with it. So as you can see here, we have a very faint band of the original mini-prep from E. coli, the Astromophilus mini-prep of around the 4,000 band, and finally, the third column shows E. coli being transformed with it after we mini-prepped the plasmid from Astromophilus. So before I hand you over to Dee, who will cover the kind of human practice side of things, so I'll just leave you with a quote that some of us working with Astromophilus wish we could have actually known about before starting this.
So, when we started our project, we wanted to understand some of the problems our project might face in the real world. To do this, we wanted to speak to some experts and get their opinions on our project and our work. So we spoke to an expert in nutrition who had experience in distributing biotechnology products in low-resource settings. He was from the Denona Institute. And he told us that our project was a good idea, as we could use dried bacterial starter cultures and distribute this to our target regions. They could then add it to their milk supply, produce yogurt, and then use any leftover yogurt to add back to fresh milk to create more yogurt. In this way, they have a sustainable supply of our product. We also spoke to a social scientist who talked to us about public concerns of GMOs and the legislation surrounding GMOs used for consumption. So he talked to us a bit about the concerns and said that we could design a self-inactivating mechanism. So together with him, we just designed and brainstormed ideas around a self-inactivating mechanism, or SIM device, that we could use to ease public concerns. So the challenges our project would face are retention of the plasmid and consumption and release of synthetic organisms. So most of the time you use antibiotic resistance to select for your bacteria. But in our case, you would have to apply the antibiotic to the yogurt and then ingest this yogurt. With the crisis of antibiotic resistance, we did not want to use this as a means, so we designed an alternative mechanism. We thought that the public would not enjoy consuming GMOs and they would be very adverse to this. So we want Astromophilus is able to survive in the gut, so we wanted to devise a mechanism that would prevent its growth and survival inside the gut. We also wanted Astromophilus to not be able to leave our yogurt environment. Using these, we thought we could ease public concerns. So we wanted to do this by modifying a toxin-antitoxin system. So this is a basic toxin-antitoxin system. It works by, it's used by low copy number plasmids and works by expression of two genes, an antitoxin and a toxin. The antitoxin has a short half-life and it degrades rapidly. It's very unstable. So you get excess production of the antitoxin compared to the toxin. The antitoxin binds to the toxin and it inhibits its function. But when a cell divides, if the plasmid is lost, you get the degradation of the antitoxin, but survival of the toxin. The toxin will persist in the cell and kill it. So we modified it in our self-inactivating mechanism. So epsilon here is the antitoxin and zeta is the toxin. Epsilon is under the, is behind a inducible promoter, and zeta is under a constitutive promoter. The inducible promoter we wanted to be activated by casein, which is found in milk and yogurt. So in this way, you only get expression of your antitoxin if you are in the presence of casein, i.e. in the milk yogurt environment. So you would not be, the bacteria would not be able to leave the yogurt environment, and when consumed, they would lose stimulation of their inducible promoter. You would get no expression of your antitoxin, and the toxin would persist and kill the cells. The results of our experiments were that we were able to get epsilon downstream of an antitoxin promoter or a constitutive promoter, but we had great difficulty cloning zeta, even without a promoter or without in a low copy number plasmid. This just comes with difficulties cloning toxins. So we were not able to submit it as a bioproduct. As I said earlier, we wanted to have an inducible promoter behind it. So in the lab, we were planning on using PBAD, but in the real world, we needed something that would be active by casein. So we were very lucky to be able to collaborate with Glasgow Polyomics, who generously agreed to perform RNA sequencing on 10 of our samples. So we had 5 samples that were S. phaemophilus in basic media, and 5 samples were S. phaemophilus in basic media plus casein. As you can see by this graph here, these are the genes that are regulated in the presence of casein. So we were going to choose one of these promoters to put in our inducible promoter construct. However, some of these genes were found to be involved in amino acid transportation, so we feel that it's probably a bit too late in the metabolism of casein, so we would need more samples and a larger time frame to be able to pick a suitable promoter. We spent a weekend at the Glasgow Science Centre to try and gauge public opinion on GMOs and our project in general, but also having designed the SIM device to ease public concerns, we wanted to see how the public would feel about our self-motivated mechanism. So we designed a small game for kids involving our mascot Beta, in which you throw ping pong balls, which are orange, to represent our bacteria, which produce beta-carotene, into our Beta bucket. And then the more orange ping pong balls they got in, the more enriched we told them that their yogurt was. While the children were playing this game, other team members would discuss GMO concerns with parents. We found that a lot of people were reluctant to consume modified organisms, but when we talked to them about our project and told them the GMOs were safe, would not survive inside them, and would not be able to escape the yogurt environment, we found that we eased some of the concerns. The major concern was the fact that the yogurt might be orange, but yeah. We don't know what to do about that. I'll pass you on to Lindsay, who'll tell you about engineering.
Great. So we also came up with a way of actually to distribute the yogurt to our target settings. So to do this, we created a piece of accessible hardware, and it would promote the idea of clean cooking. We found out that in India, milk is boiled in the morning using combustible fuels that produce CO2, and this contributes to indoor pollution. So we want to combat this by using clean cooking, and we used solar energy for this. So here's what we came up with. We used a solar oven in this case. We couldn't use a solar oven to put the yogurt into because unregulated solar ovens can get up to 300 degrees Celsius, and this would just kill the bacteria in the yogurt, so that'd be kind of useless. So instead, we used water as a heat conductive medium. The water is stored in the water reservoir at the top and flows down, controlled by a valve, into the solar oven, which we've called a solar kettle in this case, because that's essentially what it is. It heats the water up. The hot water is then flowed down, controlled by another valve, into the incubator where the yogurt will be. So the temperature of the incubator is regulated by the water. So this design can be used for pasteurising the milk at 72 degrees Celsius, and it's also used for incubation at 43 degrees Celsius. We designed our own feedback loop for this using Arduino, and the valves we used were 3D printed and designed by us, but in practice you could use any functional valve that was compatible with the servo motor. So we tested the idea one day, the only sunny day in the Glasgow summer. It was a toasty 24 degrees Celsius, 75 Fahrenheit. So yeah, we used our contraption. We tested the temperature that the water got inside it, and surprisingly, it actually reached 95 degrees Celsius, about 200 Fahrenheit, and this is more than enough for us to use for pasteurising the milk. So we're pretty happy with that. We also we needed to find out how to actually implement this design, so we spoke to a company called GoSol. They have a lot of experience in actually implementing this solar technology. They have their own product which is used in Kenya, so we were really excited to work with them. We knew that we could use their experience to our advantage, and they were really happy at what we did with our design. They only suggested that we should use an attachment, and this would make our design into a community-based rather than individual-based product. They said that this would be more widely accepted. They also said that this would be a more multifunctional idea, so not just for yogurt, which is a bit too specific for what it is. Overall, they really liked it. So to recap our project, we engineered E. coli to produce beta-carotene. We characterised the 3 Thermophilus promoters and transformed Thermophilus. We improved the characterisation of amylase P. We designed our system to prevent the growth of bacteria outside the yogurt and created a piece of accessible hardware to actually produce this yogurt. We couldn't have done this without our advisors, supervisors, and Professor Pascal Hawes and Dr. Maud Fashar, who gave us a shuttle vector, and also thank you to Exeter for collaborating with us. So, yeah, we are Glasgow. Thank you for listening. We're happy to field any questions.